In this video, I would like to explain about D-loop replication. D-loop replication. D-loop replication. This type of replication mainly occurs in the mitochondria and chloroplast. Mainly occurs in the mitochondria and chloroplast. And this is a double-stranded circular DNA. It has one heavy strand and light strand and a middle strand. Three strands are there. First one is heavy strand and light strand and one middle strand. In mitochondria and chloroplast, the DNA form a loop-like structure. And this shape of this loop is like a D. So it is known as D-loop replication. D-loop replication. This DNA form a loop and that form a D-like structure. So that is known as D-loop replication. So there are two strands. One is heavy strand or H strand and the other one is light strand or L strand, and there is a short middle strand that is formed during the replication. The heavy strand contain high adenine and guanine or high purine contents. High purine contents. Purines are double ring compounds, so it is become heavy. So it is the heaviest. The name is heavy strand because the presence of the high adenine and guanine content or high purine content. Similarly, the light strand contain high pyrimidine content. The light strand contain high pyrimidine content. Pyrimidine is single ringed structures, so it is light strand. So in heavy strand, purine content is high and pyrimidine content is less. Similarly, in light strand, pyrimidine content is high and purine content is less. So in the light strand, more thymine and cytosine are present. So when comparing these two strands, the heavy strand contain guanine and adenine and they are double ringed structures, and the light strand contain thymine and cytosine. They are single ringed structures. So when they comparing these two, the heavy chain containing more guanine and adenine, and that is heavy when comparing to the light strand. And here we can see a small fragment that is known as middle strand. And this strand acts as the primer for the replication. That is here ORI-H, origin of replication for the heavy strand, is present in this small fragment of DNA. So the replication start from here. The replication of the heavy strand start from here and it continues till 2/3 of the DNA. So here we can see ORI-L, origin of replication for light strand, origin of replication for light strand. So there are two origin of replication. One is for heavy strand, ORI-H, and the another is for light strand, ORI-L. This is the basic structure of DNA in which the D-loop replication performs. Now let us discuss how the replication occurs. What are the steps in the replication? So this is the circular DNA of mitochondria and chloroplast. Here you can see. So this is the DNA in mitochondria or chloroplast. It is a double-stranded DNA. Here you can see a heavy strand and a light strand and M strand. In this middle strand contain Uri H, that is origin of replication for heavy strand. So from here, the origin of replication, the replication starts and it continues. The replication continues and the polymerase add the nucleotides in this direction. The polymerase add the nucleotides in this direction. So that is this, in this strand, the complementary basis of the heavy strand is synthesized. The complementary basis of the heavy strand is synthesized and it continues until 2/3 of the replication. It continues and till it reaches the 2/3 of the replication. There we can see another origin, that is Oriel, that is origin of replication for the light strand. So in this place the DNA synthesis occurs in this direction. So that will be complementary to the light strand. So that is, replication starts from the origin of replication in the DNA and the complementary basis of the heavy strand. The complementary basis of the heavy strand is added in this strand. That is, this strand is complementary to the heavy strand. So the DNA polymerase added the nucleotides complementary to the heavy strand and it continues till 2/3. So at that place we can see another origin of replication, that is Oriel, that is origin of replication for light strand. So from here the origin of replication of light strand starts and that is in this direction. So we get strands like this. This is the light strand. This is the heavy strand and complementary strand synthesized for the light strand here and the complementary strand synthesized for the heavy strand here. So we get a structure like this and in this stage the ligase enzyme will come and it will join the ends of these strands. So we get two DNAs like this, two double-stranded DNA like this. One old strand and one new strand. In both contain one old strand and one new strand. So in chloroplast and mitochondria, this type of replication occurs. D-looped type of replication occurs. This is the mechanism of the replication of D-loop replication. This is all about D-loop replication.